our last discussion, we derived a very simple relationship, small p is equal to capital P plus half q, and we said that we are going to require that to extend Mendel's laws to the population level. So, let us do that in this discussion. So, let us assume that we have a diploid sexually reproducing population and we are dealing with a scenario which is a one locus two allele case. So, a trait which is determined by a single gene, and at that particular locus we have two alleles called A1 and A2. So, let us assume that the frequency of the allele A1 in parents is P. Now, from this frequency of A1 in parents, we need to go to the frequency of A1 in all the possible gametes that are going to be formed. And if certain conditions are met, if segregation is normal, equal fertility, no mutation, no migration, then we claim that the frequency of A1 remains P. Now, why is that so? Now, think about all the ways in which the frequency can differ between the parents and in the gametic stage. If, for example, when gametogenesis is happening, the segregation of the alleles is not equal, not the Mendelian segregation that we are aware of, in that case this frequency will go wrong. Similarly, suppose one of the genotypes is producing more number of gametes compared to the other genotypes. Say capital A1, let us say is producing more gametes than A2, in which case obviously A1 will be more represented in the population and the frequency of A1 P will go up. So, we need equal fertility to ensure that P does not change. Similarly, suppose there is mutation happening, again that will lead to a change in P, or suppose there is migration happening, some individuals are going away from the population or some other individuals are coming in, again that will lead to a change in the frequency of A1. And that is why in the absence of all these changes, the frequency of A1 in the gametes is going to remain the same as it was in the parents, which is P. Now, remember, in any organism, the number of gametes produced in total is way larger than the actual number of gametes which end up forming zygotes and leading to the formation of babies. Therefore, from this large number of gametes, we have to go to those gametes which are forming the zygote, and we are claiming that if the population size is large and if all the fertilizing capacity of all the gametes is similar, then the frequency of A1 in gametes forming zygotes will still remain P. Now, why are we making this claim? Remember, we are going from frequency of A1 in all gametes to frequency of A1 in a subset of the gametes, right. So, basically there is a sampling happening. Now, whenever a sampling happens, then the larger the sample and the more uniformly unbiasedly the sample has been taken, the more are the chances that the frequency in the sample is going to be the same as the frequency in the population. Now, let us look at the first part, the large sample size thing. How large should a sample be? Obviously, ideally infinitely large. Now, in order for a sample to be infinitely large, the population from which the sample has been taken also has to be infinitely large. So, that is why technically speaking we say that this is an infinitely large population. Now, similarly, I said that the sampling has to be unbiased. In other words, all the individuals in the population should have an equal chance of being represented in the sample. Now, if that be the case, then in a biological system, what does that imply? That implies that all the individuals or gametes have an equal chance of ending up being the gametes that are forming the zygotes, and that is ensured only when the fertilizing capacity of all the gametes are similar. So, for example, suppose the gametes bearing say A1 have a greater fertilizing capacity for whatever reason than gametes bearing A2. What will happen? Obviously, the frequency of A1, which is P, is going to increase. Similarly, if A1 has a lower fertilizing capacity, P will decrease in the gametes forming zygotes. And as long as the population size is very large, infinitely large, and the fertilizing capacity of all the gametes is equal, we do not expect this particular frequency to change. So, it remains at p. Now, from the frequency of A1 in gametes forming zygotes, we need to go to the genotypic frequency amongst all the zygotes. How do we go there? Now, in order to go there, we will have to make two major assumptions. What are the major assumptions? A, mating is random, and B, the frequency amongst males and females are equal. Now, why are we making these assumptions? So, if you remember, we said that the process of forming the zygotes in the next generation is essentially sampling from two gamete pools, the male gamete pool and the female gamete pool. In other words, all you can think of the process of formation of the zygotes as putting your hand in the male bag, bringing out any one gamete, putting your hand in the female bag, bringing one any gamete, and then simply putting them together. Now, suppose, okay, now if these two draws are independent of each other, in other words, if let us say you have put in your hand in the male bag and you have got an A1 gamete, does that affect what gamete you are going to get from the female bag? If it does not, then in the language of probability we say that the two probabilities are independent. Now, if the two probabilities are independent, then you have a very nice result from probability theory, which is that if two probabilities are independent, then the probability that both of them are happening together is essentially the product of the two. So, what we can do is we can essentially make what is known as the Punnett square. Okay. Let me make it here, so that I can make it a little larger and you can see it better. So, we make what is known as the Punnett square. So, let us assume that this side is the male. So, you have A1 and A2, here are my two kinds of gametes that the males are producing. This is being produced in frequency p, this is being produced in frequency 1 minus p or q, and similarly let us assume that this is the female side, this is my A1, this is my A2. Now, if we assume that the frequency of the gamete in the males and females are same, that means that this is also going to be p, and if this is p, this is 1 minus p, which means this is also going to be q. So, this is where the equal frequency assumption comes in. Now, what is the probability of an offspring being A1 A1? So, the probability is putting your hand in the male bag and pulling out a A1 gamete, the probability for which is p, and then putting your hand again in the female bag and pulling out a A1 gamete, the probability for which is again p, this p, right. So, the probability that you will form a A1 A1 zygote is the both these events happening together, which essentially means that the probability of formation of A1 A1 is p into p, which is p square. Similarly, a probability of forming a A2 A2 gamete, A2 A2 zygote is q into q equal to q square. Now, what is the probability of forming a heterozygote? Now, you can form a heterozygote either by pulling a A2 gamete from a male and a A1 gamete from a female, the probability for which is going to be qp, or you can do it the other way around, in which case the probability is going to be pq. Now, assuming that it does not matter whether you are getting your A1 from your mother or your father, these two you can consider them pq equal to qp. In other words, then the three genotypes that you are going to get, these are going to be in the ratios A1 A1 is to A1 A2 is to A2 A2, these will be in the ratios p square to pq and q square, right. That is exactly what I am telling you over here, that the genotypic frequency among the progenies will have to be this. Now, what happens if these two conditions that we are putting, these are not met? If the mating is non-random, then this nice multiplication that we are doing, that is not going to happen, because the random mating is what is ensuring that the probabilities of drawing the gametes from the male and the female pool are independent of each other, and because they are independent you are able to multiply them this way. If the mating is non-random, then this simple multiplication will not work. And what happens if the frequency in males and females are not equal? Then of course, if you have P and Q over here, you will have some other values in the female part, right. In which case again the overall logic will remain correct, but it will not be P into P, it will be P into something else, it will not be P into Q, it will be P into something else, and so on and so forth. So, as long as you have random mating and as long as you have equal frequency in both males and females, you are going to get your ratios, your genotypic frequencies in these numbers, or rather these values p square, 2pq and q square. Now, your zygotes have been formed, but all the zygotes are not going to be able to survive to become adults. So, the genotypic frequency among the progeny need not necessarily be the same as the genotypic frequency among the zygotes. However, if all the zygotes suffer equal viability, in other words all of them have a similar survivorship, which equally means similar rate of mortality, then even if some fraction is dying off, as long as the fraction is the same, these relative ratios p square, 2p, q square, these actually going to remain the same, the relative frequencies, these relative frequencies are still going to remain the same. And now remember, we have started with frequency of A1 in parents, we have to go to frequency of A1 in the progeny, right. Now, how do we go from the genotypic frequency in the progeny to the frequency of A1 in progeny? So, if you remember, I told you that small p, which is the frequency of A1 in the progeny, is going to be p plus half q. Now, here p is the frequency of the A1 genotype, which means it is equal to p square, and half q. So, q is essentially 2pq. So, this is going to be equal to half into 2pq, which basically means it is going to be p squared plus pq. Now, you can call this p dash to or p prime to show that this is in the progeny generation. So, this p prime is now equal to p, you can take this common, into p plus q, right. But q we already know is 1 minus p. So, p plus q is equal to 1, right. Therefore, this entire thing becomes equal to p, which is what I am showing you over here. As long as there is equal viability amongst all the progenies, this is going to remain as p, right. In other words, if you are starting with the frequency of A1 as p, then assuming all these conditions are met in the progeny, the frequency of A1 is still going to remain as p, it will remain unchanged, and furthermore there is a certain relationship between the frequency of A1 in the gene frequency of A1 and the genotypic frequency, and that relationship is given by this number, or rather this is the relationship which is p square is to 2pq is to q square, which essentially means these are the terms of your square of p plus q, right. This leads us to the famous Hardy-Weinberg law, which was propounded by these two gentlemen, Godfrey Harold Hardy from England and Wilhelm Weinberg from Germany. Now, this is the same Hardy who ended up discovering and mentoring Ramanujan. So, obviously, he has a very nice India connection, but I mean, although we Indians tend to think of him as Ramanujan's mentor, he also has a very important position in the annals of mathematics, in the sense that he is a very famous mathematician. Now, whatever we discussed till now, we can summarize it in the form of the statement: in a large random mating population with no selection, mutation or migration, the gene frequencies and the genotypic frequencies are constant from generation to generation, they do not change, and there is a simple relationship between the gene frequencies and the genotypic frequencies, which, as I showed you in the 1 locus 2 allele case, is given as this. In other words, the ratios of the various genotypes are the terms of this particular expansion. Now, there are quite a few comments that I need to make in the context of what we just now saw. The first comment is that, this is something that many of you are probably thinking, that isn't this a bit of a cheating? Because what we are doing is we are seeing that at every single stage, what are the ways in which a difference can happen in terms of the frequencies, and we are explicitly going out of our way to say that all the conditions are such that no difference will be there, and therefore, after all the conditions of no difference are put, what do you see? You see no difference. In other words, everything that could have made a difference are nullified, and then you get a great result saying, hey, look, allele frequency is not changing, genotype frequency is not changing. Why is that a thing? That is a very, very valid question, and the answer to that question lies in the way in which this entire relationship was discovered. So, turns out that if you remember, when Darwin came up with his theory of evolution through natural selection, the requirement of inheritance was very important, because without inheritance natural selection could not have worked. And then of course, Darwin did not have a way to explain inheritance, and that is where Mendel's theory comes in. However, in Mendel's theory you have this thing that in the F2 generation, in the monohybrid cross, you get a 3:1 phenotypic ratio. In other words, 3 parts will be tall, 3/4 of the population will be tall, and only 1/4 of the population will be short. Now, there was a particular meeting that happened, I forget where, somewhere in England, in which W.C. Punnett, a very famous geneticist, the same person on whom that Punnett square that I just now made has been named. W.C. Punnett was giving a lecture, and in that lecture, one of the people who were attending was this very famous statistician called Udny Yule. Now, Udny Yule makes a comment during that lecture. He essentially says, or at least that is what Punnett thought that he is saying, that look, if you are going to have a 3:1 ratio of the tall is to short, or of the dominant is to recessive phenotype, then over time the dominant phenotype is going to take over the population, the recessive phenotype is essentially going to go extinct. In other words, over time only the dominant gene is going to be there in the population, the recessive gene is just going to disappear. And if that happens, then remember, without variation evolution cannot proceed, without variation natural selection itself cannot proceed. So, therefore, he claimed that Mendelian theory, when you are extending it to the population, then the outcome of that is going to be that all the genetic variation on its own just disappears. Now, scholars are of two opinions about whether Udny Yule actually said this or did he mean something else. There are lots of people who think that Yule meant something else. He was a very famous statistician, he was a very sharp person. However, what is not doubted is that this is what Punnett understood. Now, Punnett was not very happy with the whole thing. He realized that there is some kind of a fallacy happening somewhere, but he did not really understand that what the hell was going on. Now, Punnett, he used to play cricket with Hardy. So, he comes back and he has a chat with Hardy, where he explains this entire thing to him, Udny Yule's objection, and says that, hey, I do not know what is going on, can you help me out here? So, Hardy, being the very sharp and famous mathematician that he was, he immediately understands what the problem is, and therefore, he immediately on the back of the envelope sketches out the proof of what we just now described, that it does not really matter whether a trait is recessive or dominant, as long as certain conditions are met, and those are the conditions that we discussed, as long as those conditions are met, the gene frequencies and the genotypic frequencies are not going to change over generations. In other words, the raw material for evolution will always be there. Now, this was a very massive result. This suggested that by itself the variation will not disappear. Only when evolutionary forces will act, only then the frequencies will change, only then evolution will happen. In the absence of those, like the law of inertia, the population will continue to have the amount of variation that it used to have. Now, Punnett himself was a very sharp person. He immediately understood the implication of this entire thing, and he strongly requested Hardy that please write this up and publish. However, there was a problem. What was the problem? Problem was that Hardy considered himself to be a quote unquote pure mathematician. In other words, he was of the opinion that mathematics should be done only for the sake of it. Mathematics should not be done so that it leads to a benefit, or it ends up proving making some applied contribution. Therefore, Hardy was extremely reluctant to get this thing published, because he thought this is an absolutely trivial result, and more importantly it will make life useful for somebody, the biologist, which obviously he thought was beneath his station. But Punnett ended up pestering him, and finally at some point Hardy relents and he writes his paper up and ends up publishing it in the Science. So, this is a very famous paper, just one page, and it starts with this absolute brilliant thing. I am reluctant to intrude in a discussion concerning matters of which I have no expert knowledge, and I should have expected the very simple point which I wish to make to have been familiar to biologists. So, you can see the innate bias of or innate superiority complex of being a mathematician showing out. Then he goes on to say, however, some remarks of Mr. Udny Yule, to which Mr. R.C. Punnett has called my attention, suggest that it may still be worth making. And then he goes on to sketch that proof. Now, this is the sole contribution that Hardy makes to the field of biology. And very interestingly, of course, Hardy, if you talk to the mathematicians, they will tell you what Hardy has done. However, outside the world of mathematics, Hardy is actually way more famous for making this certain contribution in the context of the Hardy-Weinberg law, which is a big irony given that how Hardy would have completely hated the fact that his work made some applied or important application oriented contribution. Anyway, Hardy writes his paper in English, as you can see, and therefore, this becomes reasonably well known in the English speaking world. However, around the same time in 1909 actually, Wilhelm Weinberg completely independently ends up discovering the same principle. In fact, he ends up doing a lot more work related to this than Hardy ever did. I mean, Hardy did nothing, he just published this paper and he stopped. Weinberg does a lot of work, but unfortunately Weinberg was publishing in German. That is why his contributions were not known to the English speaking world for quite some time, and at some point when it became clear that Weinberg figured it out on his own and did a lot more, at that point it was proposed that the names of Hardy and Weinberg should be put together, both of them should be given credit for this discovery, and that is how we got the Hardy-Weinberg equilibrium. Now, this statement is the full statement of Hardy-Weinberg equilibrium. However, it is my observation over the last several years that students somehow do not think of this statement as Hardy-Weinberg equilibrium. So, whenever in the interviews etc., I ask people what is the Hardy-Weinberg equilibrium, then they simply say Hardy-Weinberg equilibrium is p plus q equal to 1 and p square plus 2 p q plus q square equal to 1. Now, the problem with these two statements are that they are correct. In a one locus two allele scenario, the genotype frequencies and the allele frequencies will sum up to 1, there is no doubt about that. But the point is, you do not need two great mathematicians' names to be associated with a result if that were only that trivially true. And that is why I tend to sometimes ask students that you are saying that p plus q equal to 1, p square plus 2 p q plus q square equal to 1. Why do you think the names of two great people are associated with such a trivial result? And you know what the answer that I get? So, one of the, well, I get many answers. One of the major answers that I got was yes sir, I fully agree it is a trivial result, but you see even a trivial result needs to be thought the first time, and these were the two people who thought about it the first time, which is why it's their names are associated with it. This incidentally, as you can see, is not correct. This is not the statement of Hardy-Weinberg. This is what Hardy-Weinberg stands for. The other very important thing to note is that if this is all that is there to it, this statement, then why is it that I am showing you this entire derivation? Why is it that I am pointing out where each and every one of those assumptions are impinging upon? The reason for that is that by itself the Hardy-Weinberg law is absolutely uninteresting. As I said, that the Hardy-Weinberg is simply saying that if all the ways in which allele frequencies can change are blocked, then allele frequencies will not change, right. So, in that sense the law itself is not interesting. What is interesting is the fact that in real life it is very rarely that a population is going to meet all these assumptions that I have written over here. So, if that be the case, the entire subject of population genetics proceeds by figuring out in what way all these assumptions that I have written over here, in what way are these assumptions being violated. So, in that sense the Hardy-Weinberg law is a null law. It is equivalent to Newton's first law. Okay. What does Newton's first law say? It says that a body is going to remain in its state of inertia, either moving or static, whatever, until and unless an external force acts upon it, right. So, Hardy-Weinberg also says the same thing, that a population is going to maintain its variation until and unless an external force, evolutionary force, acts on it, and therefore, the whole idea of population genetics, the whole goal of population genetics is to figure out which one of these forces are acting and in what way are they acting, what are the relative contributions of the various forces, and so on and so forth. So, that is why this is one of the few laws in biology. Remember, there are very few laws in biology. This is one of maybe 4 or 5 that exists. So, there are a few points to be noted about the Hardy-Weinberg equilibrium. First point, which is a part of the statement itself, is that the gene and the genotypic frequencies, they do not really change from generation to generation. The second point is that the relationship between the gene frequency and the genotypic frequency is reached in a single generation in the one locus case. This is very important. So, what do I mean here? So, suppose you have the population which is in P. I know the allele frequency of one of the alleles is P, which means that its frequencies, genotypic frequencies, are P square, 2 P q and q square, right. Now, suppose for whatever reason this becomes the allele frequency of the first allele becomes P 1, in which case you expect the genotypic frequencies to be P 1 square, 2 P 1 q 1 and q 1 square, right. So, the question that we are asking is how long will it take for the genotypic frequencies to go from P square, 2 P q q square to P 1 square, etc., etc. Now, turns out that if, as I said, if it is a single generation case, single locus case, then it will happen in one generation. However, even within the single locus case, if the frequencies of the alleles are different in the males and the females, then it will end up taking more than one generation. And when we go to the two locus case, then it typically takes way more than one generation, depending on what is the recombination frequency between the loci and depending on something known as the coefficient of linkage disequilibrium. Of course, when you are talking about two loci or more than two loci, then things like epistasis etc., also come into picture, which of course can make this relationship genotypic frequency relationship never being attained. But that is a way more complicated case, something that we are not going to consider as part of this particular course. So, third point is related to progeny genotypic frequencies, them being dependent only on the parental gene frequency and not on the genotype frequency. Now, what do I mean by this? What I mean is that, remember, our small p is equal to capital P plus half q, right. So, in principle you can get the same value of small p by altering the capital P's and the half q's. In other words, different parental genotypic frequency can lead to the same parental gene frequency. And what this statement is insisting is that in the offspring generation, the relationship between the gene frequency and the genotypic frequency depends only on the gene frequency of the parent, not on the genotypic frequency of the parent. What do I mean here? So, let me give you an example. Suppose I have a population in which my A1A1, A1A2 and A2A2 are in these frequencies 0.3, 0.6 and 0.1. And let us assume that in another population the frequencies are 0.2, 0.8 and 0. We do not have any A2A2 at all. Now, what are my gene frequencies in these two cases? In the first case, this is 0.3 plus half of 0.6, which is 0.3, which means 0.6. In the second case, in this case, this is 0.2 plus half of 0.8 is 0.4, which means my gene frequency is again 0.6. So, in both these cases the gene frequencies are same, and therefore, when it comes to the genotypic frequency in the progeny generation, in the next generation, in both cases it is going to be 0.36 is to 0.48 is to 0.16, which is simply 0.6 squared and so on and so forth. So, this, if you want, you can actually simulate it for yourself and convince yourself that that is how it works. Excellent. So, now there are two further points about the Hardy-Weinberg equilibrium, but before we get to those, I need to introduce you to this diagram. So, what am I doing over here? So, here we are representing the alleles as capital A for A1 and small a for A2, and here the frequency of capital A is P, which is going from left to right. So, as you go from left to right, P is increasing, which obviously means that the frequency of the other allele small a is increasing from right to left. And these three lines, they depict the genotypic frequency of A1A1, A1A2 and A2A2. So, this one is for I am sorry, capital A, this one is for small a, and this one is for the capital A, small a, the heterozygote. Now, let us first concentrate on the first homozygote. As you can see, when the allele frequency P is very low, then the homozygous frequency is very less, but as P increases, this increases quite fast, which is obvious because this is going by the square, and then when this frequency of allele becomes 1, the frequency of the homozygote also becomes equal to 1, no issues. The opposite happens for the other allele. When the frequency of that allele is very high, when it is at 1, then the frequency of the homozygote is 1, and as the frequency of the small a allele keeps on decreasing, then this frequency of the homozygote also keeps on decreasing, and when this frequency becomes 0, this frequency also becomes 0, no issues. The interesting thing is that for both the homozygotes, the maximum is when p equal to 1 or when q is equal to 1, this point for q equal to 1 and this point for p equal to 1. But when it comes to the heterozygote, the capital A small a genotype, then you can see that the maximum value of that is occurring somewhere in the middle, and for the 1 locus 2 allele scenario, this value is going to become maximum when p is equal to q is equal to 0.5. And what will be that maximum value? Simple, 2 into 0.5 into 0.5. So, 0.5 into 0.5 is 0.25. So, this is going to be equal to 0.5, and that is what you can see over here. Okay. So, this is 0.5. So, I am not demonstrating this to you, but if you know very simple calculus, you should be able to show using the first derivative and the second derivative that the maxima is indeed this point and the value of the maxima is 0.5 and this maxima occurs when p equal to q equal to 0.5. Okay. So, that is what I am telling you over here. The next point is a slightly more subtle point and that point is related to the relative frequency of the homozygotes and the heterozygotes. Now, look at some point over here. Okay. This is where p is 0.1 and obviously p square is very small, it is 0.01. However, even at this point you can see that this value of the corresponding heterozygotes is actually reasonably high, it is close to 0.2, right. So, this tells us that when an allele in the population is in Hardy-Weinberg equilibrium and when that allele is rare, then most of the copies of that allele is actually going to be inside the heterozygote, right. Now, why is this a worthy point of making? It is worthy because of how human, or not all, but most human diseases behave. So, if you look at most diseases, well, not only in humans, in many animals actually, most of those diseases, genetic diseases are caused by recessive alleles. Now, obviously, if a disease is caused by a recessive allele and that disease is leading to some problem in terms of survivorship or in terms of how many babies are being produced, then you expect natural selection to take away those alleles from the population. In other words, you expect natural selection to reduce the frequency of those alleles. Now, obviously, natural selection has been acting for many years and therefore, you expect that over time natural selection will completely be able to weed out those alleles from the population. Yet, many of those diseases are very much there. How does that happen? Now, there are multiple ways in which it can happen, but one of the ways that we have to remember is coming from this diagram, which is that natural selection can push the frequency of that allele to a low value. But when it does so, the copies of that recessive allele, most of them they end up in the heterozygotes, and when they end up in the heterozygotes, they are not being expressed because they are recessive, right, and at that point natural selection cannot do anything against them. And that is why for natural selection to weed out a recessive allele through selection alone, it becomes a very difficult proposition once the allele has gone beyond a certain threshold. And this is one of the reasons for which many recessive genes are still, or recessive alleles are still present in the population. So, that is the point that I am making over here. So, before we wrap up, a quick note on why the Hardy-Weinberg equilibrium is such an important thing in biology. So, the first point is that this is one of the few laws in biology, and as I said, by itself it is hardly of any interest, but the entire subject proceeds by looking at the departures from the law. In that sense, it has the status of a null law. It is like the first law of mechanics, Newton's first law of mechanics, which says that a body will continue in its state of inertia until and unless acted upon by external force. Again, there is no body on which some force or the other is not acting, and therefore, the law by itself makes no major prediction, but the entire field of mechanics actually proceeds by asking how is the law being violated and in what ways forces are acting on the body such that the state of inertia is being violated or is being changed. Exactly the same thing happens in the context of this particular law. You looked at the assumptions, right? There are so many of them, and there is hardly any population which is going to obey any or all of those assumptions. Somewhere or the other the violations will happen, and when the violations will happen, there will be a departure from Hardy-Weinberg, and the entire subject of population genetics is trying to figure out why that departure is happening, which of the assumptions are being violated and to what degree. And of course, in its purest form, this law is suggesting that in the absence of any evolutionary force, allelic diversity is going to be maintained. This itself is a very big thing, because this suggests that the raw material for selection to work, for evolution to work in fact, is being, all else being equal, will be maintained on its own. It is not that it is going to, allelic diversity will go away and therefore evolution will stop. And in that sense, this is a very strong supporting thing for Darwin's theory of evolution. And although we are not showing it to you over here, but this law actually expands very easily to both multiple alleles and multiple locus cases, multiple loci cases. When it comes to multiple alleles, it actually is very simple. So, for two alleles, the relationships, the genotypic relationship are terms of this expansion a plus b whole square. If you have three alleles, it becomes the terms of the expansion a plus b plus c whole square. If you have n alleles, it simply becomes a plus b plus c plus dot 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 up to n whole square. Okay. So, the terms of this expansion become the various terms of the genotypic ratios, right. In the context of multiple loci also, it is relatively easy to take it over there. Of course, it becomes a slightly bit more complicated, the maths becomes slightly bit more complicated, but it is still tractable. And then finally, from a very application point of view, this helps in letting us know what is the frequency of the carriers of disease in a population. So, who are the carriers? Carriers are the heterozygotes. Now, if you are a public health person, you need to figure out what is the rate at which rare diseases, relatively rare diseases are going to surface in the population. And I mean, obviously, this is needed so that you can take appropriate measures for treating them in the hospitals and so on. And in order to understand that rate, you need to understand what is the frequency of the heterozygotes. And as we are going to show you in the next discussion, you can do that by assuming Hardy-Weinberg and then doing some relatively simple calculations. So, that kind of simple calculation and a few others, that is what we are going to do in the next discussion.